Start your presentation anytime.
All right, great. Well, hey, I'm Kevin Liu. For those of you who I guess haven't had the pleasure yet of meeting, yeah, I'm a faculty member in the Department of Computer Science and Engineering. It's really been my great privilege to be involved with Beacon over the years. So I've been here for a few years now. Um, and what I wanted to chat with you all about today was one of our latest projects, which is, well, the applications are so far we've been applying it actually to a few different areas within evolutionary biology and just kind of computational biology and bioinformatics, but it's kind of more general than that. And so I'll kind of give you the bigger picture of this in just a second, but the talk itself, it's dealing with this issue of trying to assess statistical confidence, um, and really trying to use some pretty basic statistical approaches to look at fundamental issues with rigor and reproducibility. That's basically it. And by and large, we're only looking at one topic for today, which is just biomolecular sequence analysis. But I really want to start with, the people who honestly are really doing the work. And so I just wanted to highlight some of the people who have made major contributions to this project within my lab. First off, we've got my PhD student, Wei Wang, an amazing PhD student who has done actually a lot of cool stuff. And she will be defending her thesis within this academic year. So if you're interested in potentially working with somebody as maybe a postdoc or something like that coming up soon, feel free to reach out to me. I'd be happy to put you in touch with her. Yeah, she's just a very talented individual who really has been kind of honestly leading this project. And at multiple points during the project, she really kind of broke through major barriers. So I honestly have to say, yeah, Wei, she is one of the major people who's driven forward this project. In addition to that, we've had major contributions from a really spectacular undergraduate student, Jack Smith, who unfortunately I could not convince him to come do PhD studies in any capacity. He just, the way he put it was he got an offer that he couldn't refuse from Microsoft, which I interpreted to mean, a big offer. Very happy for him. He's really happy in Seattle now. And finally, my most recently graduate PhD student, Hussein Hujazi, who recently took a position with Janssen Pharmaceutica, also an amazing student who's done a lot of great things. It just turns out that just this year, one of the things that was pretty nice that happened for me is his brother actually joined my lab. So now I have the privilege of advising his brother, Ahmad, who's working on kind of related stuff. So at the very end, some of the future directions that I'll briefly hint at, actually Ahmad's been working at. So what is our project about? As I hinted, we really wanted to address a fundamental issue related to looking at rigor and reproducibility, but specifically within biomolecular sequence analysis. And throughout really any place where you try to apply computation to the biological sciences, I would say that sequence analysis, you'll often see it appearing in a lot of different ways. So some of the more classical problems include things like multiple sequence alignment. If you have a set of bimolecular sequences, odds are, depending on what you're looking at, they can be of different lengths for a variety of different reasons, including very important biological reasons, such as you'll have sequence insertion and deletion processes that happen over time as evolution occurs. So this is one of the reasons why you need to try to align data and figure out what exactly within your sequences are homologous or not. So this is a really classical task, and that's really one of the core tasks that we'll focus on today. It's also been researched for quite a long time, many decades at this point. Some of the other problems in which you'll see biomolecular sequence analysis crop up are things like trying to reconstruct phylogenies or evolutionary trees and other more general graphs and other structures like that. Trying to do genome annotations and lots of other areas as well. So this is just a really brief bulleted list of things where you could try to apply topics where you're looking at trying to assess and quantify rigor and reproducibility in the context of biomolecular sequence analysis. Now, like I said, this is a really big topic. So here's my shot at just giving kind of a high-level summary of where this fits into the bigger picture. I would say that really anywhere you're trying to apply statistical models to do model-based inference and learning, you could in theory try to apply these common techniques for assessing and quantifying rigor and reproducibility. So here's one picture of what that might look like such a framework. You've got typically some kind of unobserved natural process that you want to study, for which you actually only get a limited set of observations, typically, usually, dealing with only observations that you can make pretty much in the present day, especially for the topics that we'll talk about today. Really only the extant present day organisms or systems can you actually get sequence data for. And so your whole goal is you want to take basically this limited set of observations, typically about the present day, and try to reason about what's happening but unobservable to you, okay, this natural process. And so a common approach is you try to adopt a reasonable mathematical or statistical model of natural process under study. And then using that model, you try to perform inference and learning, usually using some sort of computation to do so. And this is this so-called model-based method to do inference and learning. And this is the way in which you try to make estimates or reason about things that are otherwise unobservable to you for this natural process that you might be interested in studying. And so you can do this, and people do this commonly, and and not just the fields that I work in, I'm sure the fields that y'all work in. But by and large, I think as scientists, a basic kind of fundamental challenge is we should be skeptical, right? And you should think carefully about, okay, you can run a method, but to what extent should you actually trust the output that it gives you? And I would say it's probably something that is a very well accepted idea, and yet in a lot of studies, I would argue that there's plenty of room for more improvement. Let me just put it that way. And so one of the ways in which we could do better, I guess, or just this issue could be addressed better, is trying to adopt quantitative approaches to try to assess repeatability. And so one of the ways in which you could do that is, ideally, if you could somehow get more data somehow, then what you could do is whatever model-based inference and learning method that you're trying to use, you could simply rerun it on this data if it were accessible to you and just see, well, okay, it gives you an output. Is it going to give you the same output or something different? And so that's the basic idea behind trying to assess repeatability, where you could try to use resampling to help you do so, okay? And so the ideal case, here's a little bit of an illustration of what I just outlined, right? Like ideally, in theory, if you have this process and somehow if you could get a bunch of different replicates, so sample a bunch of different kind of replicated data sets from your process, you could repeat your method, repeat the analysis that your method does on each of these different data sets, and simply just compare across the different re-estimates and see to the extent do your re-estimates agree or disagree, okay? And you could propose different quantitative measures based off of what agrees and what disagrees out of your output re-estimates. And so this is the theoretical case, but typically we aren't so lucky that we have just tons of data that we have accessible to us. There can be costs associated with that. Or furthermore, just other expenses like, maybe it's just risky to get more data, et cetera. Or we simply just don't have access to this process at all, or there's no reasonable way to just get that data in any easy way. And so whatever data you got, that's kind of what you have, okay? And so what do you do in this case? Because in practice, this ideal case really it's not always kind of a practical thing that you can rely on. So instead, there are really, I would say, at least two common classes of approaches for performing resampling and re-estimation, rather than just trying to get more data from your process under study. And so one of the ways in which you could try to actually get more data is using a so-called parametric resampling approach, which simply leverages this idea that if you're using a statistical or mathematical model already for doing inference and learning, if that model is generative, then you could simply just use that and just get more data from it. So just resample more data from the statistical or mathematical model that you're using for inference and learning. So here's a picture of this scenario where you're using your model-based method on your original data, and then everything kind of on this side is just the same picture as what I've just introduced. On the right side is you're using this same model that's driving your inference and learning, but then simply just drawing more data sets from it, right? And this is typically for the types of models that we'll talk about and that are often used. It can be the case that it's actually pretty straightforward and practical to resample from them. So you can like write a computational procedure, write up some software to do this simulation or resampling process, and we'll actually use that to study the methods that I'll talk about today. And so in this case, then you get lots of data, as much as you want, and you can then do your re-estimations on your parametrically resampled data, and then do this comparison that I just talked about. And that's another way of trying to quantify agreement or disagreement across re-estimations. Now, this is a common approach. I will point out that it has this fundamental reliance on the statistical model. And so, all along I've been talking about model-based inference and learning. There's this almost cliche now, people talk about how, models, they're not perfect. Everyone knows that. And so, there's models which are used, but everyone accepts that no model is perfect, right? And so to some extent, your reliance upon a model, especially relying it not only for inference and learning, but also to drive the resampling and re-estimation procedure, it's just more and more reliance on a model. And so trying to deal with approaches which don't use explicit parametric models to drive the resampling and re-estimation, they're also really widely used and really popular. One of the chief advantages being they don't rely on a parametric model in the way that the parametric resampling approach does. And so here is that picture updated now with an alternative for non-parametric resampling, where you've just got some method where typically the way that it works is, instead of trying to use a statistical or mathematical model and trying to draw more data out of it, you try to somehow manipulate or kind of augment your data by kind of perturbing the original input data that you're given. Okay. And so that's typically how these non-parametric resampling methods work. They perturb or manipulate the original data sets into new resampled replicates that are supposedly representative of the original process, right? It's a proxy. It's almost a workaround, but I would just say the better way to put it is it's a proxy to drawing more data from the natural process. If you can do this perturbation to the original data to get more resampled replicates. And then everything else after that point is the same as the picture that I've been building up so far, okay? And so these two classes of methods are widely used, but arguably of the two classes, I would say probably the non-parametric methods are some of the most widely used ones, simply because they don't have a reliance upon, you don't have to assume that a statistical model has necessarily generated your data, right? You're just taking your data and then somehow applying a non-parametric method to get more resampled replicates from that data. And out of this class of non-parametric resampling methods, by far one of the most widely used such non-parametric resampling methods is the so-called standard bootstrap method, which is pretty simple. It just does the following. If you've got an input sequence of observations, you simply just sample uniformly at random from your input set with replacement. That's the key. So just take random observations uniformly at random, take one at a time, and then after you've taken that out, written it down, then put it back into your input set, and then do that same process again, and keep going until you feel like stopping, right? Until your resampled replicates sampled, and you that's it. That's basically it. Uniformly random resampling with replacement. So this whole process, it has a really important assumption, which is that you can indeed sample from your input observation sequence independently, sample independently each observation. And furthermore, it assumes that they're identically distributed. So this is making this assumption that your data are independent and identically distributed, or i.i.d. And so this is a reasonable assumption in certain conditions, but unfortunately for biomolecular sequence analysis, there are actually a lot of different reasons why your data are inherently sequential in nature. And you really should try to account for why one observation coming after the next, maybe a nucleotide or something like that, in the case of nucleotide sequences in like a genome or something, the fact that one nucleotide occurs next to another, that actually is really important to capture for a lot of different reasons. And some of these reasons include things like biomolecular structure and function. Maybe you're looking at protein sequence, amino acid sequences or something like that, which actually have structure. Maybe there's some kind of, I don't know, biomolecular function to the region of the genome that you're trying to analyze. There's also other evolutionary processes that basically cause neighboring sites within a biomolecular sequence to basically evolve together and be linked. Things like genetic recombination and also sequence insertion and deletion processes during biomolecular sequence evolution. And so in each of these cases, trying to assume that your data are i.i.d. or independent and identically distributed, it just doesn't make sense because of these fundamental reasons. So kind of based on that, what our lab tried to do was we came back to this idea that trying to do non-parametric resampling, it's really useful for assessing reproducibility, but we'd like to try to avoid this pitfall of assuming that your data are i.i.d. We want now instead a non-parametric resampling procedure that can preserve the sequential dependence of the data that you're given. And so what we ultimately did was we created such a new method which integrates not only ideas from Bootstrap Resampling, but a really clever idea due to Landon and Grauer back in 2007, which is their heads or tails method, which basically says the following. It's a pretty intuitive thing. It says, if you're doing the procedures that we've been talking about, right, doing biomolecular sequence analysis of any kind, you do your analysis, great, you get some kind of inference or estimate. It should be the same whether or not you read your input sequences from left to right or from right to left, right? It's pretty intuitive. And in a lot of different cases, that's absolutely, your expectation. And so this is basically kind of mirroring your inputs and using that to do some limited resampling and re-estimation. And so we tried to basically build on these ideas, and that gave rise to a new non-parametric resampling method, which we refer to as sequential resampling or series. And at a high level, what the series method is it's basically just trying to do essentially a random walk on your data. That's basically all that it is. And so thinking about this idea of trying to mirror your inputs, reading your sequences left to right, doing your analysis, and then comparing that to reading your input sequences from right to left, doing your analysis, right, and comparing those two estimates. This is kind of that same idea, except that you're trying to get more replicates now by basically switching up how you're doing that reading. That's essentially it. And so to actually do this, though, on input sequences where you can have them of potentially differing lengths, you have to be a little bit careful. And so there's some additional kind of complexities that arise. And so I'm going to walk you through that now with just a little bit of an illustrated sketch of how this procedure works. So here on the top is a little example input with 5 different, in this case, nucleotide sequences that are part of our input, where they're of different lengths. And so you can imagine that, among the different problems in biomolecular sequence analysis that have inputs that look like this, some classical ones are things like multiple sequence alignment, okay? So given this input of 5 unaligned sequences, the multiple sequence alignment or MSA problem, it just says, okay, give me an alignment where you're putting it into this matrix by adding in indels or dashes to line up your nucleotides, where having a nucleotides lining up within a column, basically that talks about sequence homology, right? And so such an output could be something like maybe the bottom here. And so the very start of this series procedure is we need some kind of rough estimate of what an alignment might be, but only for one thing, which I'll talk about in a second. So we need a rough kind of guide estimate of what the alignment is. And so you could do this a few different ways. Within our initial study, we actually took a really kind of rough and ready approach to this, just picked a multiple sequence alignment method and used that. You could also try to take a consensus of a few different MSA methods and use that. but you're really using this only for one purpose for resampling, for series resampling, which is you kind of need some place for your random walk to synchronize. That's basically it. Because otherwise things could get really kind of desynchronized and start to get ugly. And so the way in which we do this is we don't care about most of our initial guide alignment. We only care about the most reproducible, the most conserved parts. And so basically out of your initial alignments, we'll only focus on the very few short regions where they are most conserved. So in the case of the consensus alignment, you could use a few different approaches for trying to assess this. We did the simplest thing possible, which is just look at where sequence identity is highest. And so for these short regions, in this little illustrations, they'll be very short, something like just 2 columns long, whatever. These form a set of synchronization points, which we refer to as barriers. And so all that they're used for is basically forcing the random walk to synchronize at those points. But otherwise, you basically read the input sequences in an asynchronous fashion in between a pair of barriers. So for example, here is the start of such a random walk. Where you're walking from one barrier to the next. Okay, so I've been using this term barrier, which really just refers to kind of the edges of these conserved regions. So you walk from barrier to barrier, as I just said, and in between each pair of barriers, you're always reading the sequences in the same direction, but asynchronously. So they could be of different lengths. So in this particular case, it just turns out that they're of the same length. That's fine. For this initial step from the first barrier that you randomly selected, you picked a random direction for your random walk and just started walking to the next barrier. In between, you just read the sequences. And so this process continues, but I'll point out that at the next, so from the second barrier that you hit to the third one that you hit, you're actually reading your 5 sequences, but it turns out that they're of differing lengths. And so here, in the next parts of the sequences that you're resampling, they're of differing lengths. So in S1 you get TAAT, but then in S3 you get all this extra stuff: TAAT, GG, GT, GGG. So you'll see all that stuff appearing here in your resampled S3. Okay, and this whole process continues until kind of like Bootstrap Resampling and just kind of related procedures until you've sampled enough data for your replicate. And so we use a simple length criterion to basically say when our resampling process has finished. And so in our study, we use a simple criterion of just such that the resampled data is at least the same length as the original input data. So yeah, in this particular case, you conduct it's just a completely random walk with some probability for reversal, which is a method parameter. And it turns out that, well, you keep going until you hit the end of the sequences. And then at the end of the sequences, you always have a certain reversal. Then here, I guess at this barrier, it turns out that there was a random reversal that happened. The random walk just reversed, went back in the other direction, and then finally the length criterion was satisfied when you hit the barrier at this point, and that's it. Your resampling is done, and you'll have one resampled replicate data set as shown. Okay, and so this is just a little bit of an illustration to give you an idea of what the random walk actually looks like when you conduct this series procedure. Okay, and this is how you resample your biomolecular sequences of potentially differing lengths using such a random walk procedure. And it's all it's trying to do is combine this idea from Landon and Grauer, right? Mirroring your inputs. It shouldn't matter whether you read left to right or right to left. But also with this idea that you can start from different places and you can also let that right to left versus left to right reading kind of change up during your resampling. Okay. Where does it go? And so this is the resampling procedure. And what we then wanted to do was try to study whether or not it's useful, because, hey, the Bootstrap method, it's used widely for a lot of different problems. But by itself, it's just a statistical procedure. Its utility comes from, well, in part from its applications. And so in the same way, we wanted to study the application of this random walk-based resampling procedure to a specific inference or learning task, which in our case, we selected one of the classical problems that we just talked about, that of multiple sequence alignment estimation. And we tried to see, okay, if you do this procedure to try to get more data and then redo your alignment estimation, does this help in any way? And so this problem of trying to assess and quantify reproducibility for multiple sequence alignment estimation, it's actually pretty well studied. I'll give it this particular name, MSA support estimation, just to be clear. I don't care about like trying to align trying to estimate one multiple sequence alignment on one input. I'm trying to assess reproducibility for a given multiple sequence alignment estimate. Okay, it's related, but not exactly the same thing. So this problem I'll refer to as multiple sequence alignment support estimation. And what we set out to do was to study whether or not this particular new resampling procedure, whether or not it's useful in the context of MSA support estimation. And so our performance study looked at this question using, well, first off, simulated data. And so there's a simulation study component to evaluating performance, which I'll talk about briefly here. It involved a range of data set sizes between 10 and 50 taxa. Sequence evolution was simulated under a sequence evolution model, which incorporated a finite sites model of nucleotide substitution, as well as a particular indel model taken from a 2012 study, an earlier 2012 study. I guess one of the more important things to note, it did capture a range of, I guess, difficulty is the way you call it, due to how divergent or not the sequences were. And so there was a range of model tree heights, which basically captured or resulted in a range of evolutionary divergence for the simulated sequences under study. And so we named the 10 taxon model conditions 10.A through 10.E in order of increasing model tree height and therefore increasing sequence divergence. And similarly, you'll see there's also 50 taxon model conditions named 50.A through 50.E, just in the same way. Okay. And so this is just the simulated data that we used for our performance study. There was actually a companion set of empirical benchmarks that we used as well, which if you're interested in, I'd be happy to chat with you, and or you could just take a look at our publication as well to find the details for that. So as I had said, this problem of MSA support estimation, it's been studied over the years. And so to date, some of the more popular state-of-the-art methods include methods such as the first guidance method from Penn et al. In 2010, and its successor method, guidance 2, which it incorporates a lot of different ideas to try to basically do the same process of trying to get more resampled data, but in a different way than what we're doing. Using our approach. And so our approach, it's fundamentally using this random walk resampling procedure to get more perturbed data, kind of in the same way that the Bootstrap does, and then trying to basically use that to drive reestimation and thereby assess reproducibility. I should note also that technically speaking, we did basically combine this idea with some of the ideas from guidance 1 and guidance 2. And so in the results I'm going to just show you in just a minute, basically the series-based methods are therefore referred to as series plus guidance 1 and series plus guidance 2. It's really trying to combine these ideas. Okay, I'll briefly mention just the different performance measures that we utilized in our study. They include things that measure common measures of type 1 and type 2 errors, such as using receiver operating characteristic curves and precision recall curves. And so the way to think about it is, for example, in receiver operating characteristic curve, it's just a curve that plots true positive rate, which is true positives divided by true positives plus false negatives on the y-axis versus the false positive rate on the x-axis being the false positives divided by the false positive plus true negatives. And it's just trying to assess type 1 error versus type 2. And so in a sense, it's kind of, you can trade off basically type 1 versus type 2 error to give you different points along this curve. And kind of a similar thing is going on with, different measures being precision and recall in the case of a precision recall curve. And so to get kind of a single measure of type 1 and type 2 error, you can try to look at area under curve to look at this trade-off in each of these two cases. And so I'll show you results I think it's for area under precision recall curve, just to give you an idea. But basically the idea being more area is better, i.e. there's less trade-off that you require. And so in general, yeah, I guess the way to interpret this is there's just better type 1 and type 2 error overall, where you have more area under curve. The other measure that we'll look at briefly is looking at the computational runtime and main memory usage of the methods that I just mentioned. And so here are just a little bit of the results coming off the simulation study. I know it's a lot of numbers, but just let me walk you through the layout of this particular sort of messy table. It's reporting that one measure I briefly mentioned, which is area under precision recall curve, measured as a percentage between 0% and 100%. As I had said, bigger is better. So 100% means the littlest trade-off possible, i.e. a perfect method that has perfect type 1 and type 2 error. So let me see. The other thing I should orient you through in this table is the rows, which basically correspond to the different model conditions in our study. As I said, we have 10 taxon model conditions named 10.a, 10.b, up until 10.e in order of increasing sequence divergence. Okay, so basically 10.e is most divergent. You might guess that it could be hardest for that reason. And 10.a is least divergent. Like all the sequences look most similar because of least model tree height, least model phylogeny height. And so that's the first 5 rows that you'll see here. And then the last 5 rows are for the 50 taxon model conditions, which are named similarly. 50.a is least divergent, easiest, I guess. And then 50.e is most divergent, most difficult. Okay. And now in this table, I'm comparing guidance 1, the earlier MSA support estimation, which is still somewhat widely used, versus a pipeline, which the most important difference is it utilizes series for driving the resampling off of your input sequences to get more resampled sets of unaligned sequences. There's a little bit of extra stuff that happens, which just incorporates ideas from guidance 1. As I said, this is why I refer to it as series plus guidance 1. And so across all model conditions, when you're looking at aggregate area under precision recall curve, in all cases, we see improvements of the series-based pipeline versus guidance 1. That's the first thing to note. And so basically, I bolded the best of the 2 methods for this reason. And the improvements, if you kind of stare at the numbers for a second, they actually increase somewhat as you go from least divergent to most divergent model conditions. So for example, from 10.a versus 10.e, and then equivalently the 50.a versus the 50.e model conditions. The improvement in type 1 and type 2 error tends to increase as the sequences get more divergent, and I guess the MSA problem becomes more difficult per se. And then also just doing a basic statistical test to just verify statistical significance, and also incorporating a correction for multiple tests, you do find that these improvements are statistically significant as well. Here, that's the performance results for type 1 and type 2 error for the guidance 1 versus series-based equivalent series-based pipeline. Here's the same set of results for guidance 2. In the interest of time, I'll just cut to the chase. Kind of same results, but a little bit less improvement because overall guidance 2 is more accurate in terms of type 1 and type 2 error versus guidance 1. But you do see consistent improvements by applying series-based resampling re-estimation in this performance comparison as well. And those improvements are also statistically significant. Briefly, here are results regarding runtime and main memory usage for all the methods that I just mentioned. In brief, runtime here is shown on this top panel. The runtime in hours is the y-axis. The x-axis are the same list of model conditions running from left to right. And the methods here are shown. Long story short, every single method analyses took roughly on the order of half an hour, maybe just, well, under an hour, let's just say, under half an hour. There were some differences, I would say. So there is some overhead that's imposed by this resampling procedure. Critically, our approach does require re-estimation of the alignments, whereas, oh, shoot, sorry, I'm thinking of something else. That's a misstatement. So in this particular study, basically, there is a little bit of overhead added by the series-based resampling procedure. And that's basically represented in kind of this pretty small differences shown here. But I guess I could have zoomed in here to exaggerate the differences, but they're on the order of minutes, I guess is the fair way to assess this. And overall, the time that you need to assess reproducibility using any of these approaches, it's on the order of minutes. So very, very, feasible. Not a lot of overhead there. Main memory usage as well. Main memory usage is easily tractable, at least for these synthetic data sets, very tractable by modern desktop PC standards. We're talking of, just a few gigabytes of main memory usage required at most. So, this particular laptop that I'm using is perfectly capable of running these types of MSA support estimation analyses for any of these methods. There is, again, overhead that you see that's added by the series-based methods. That is important to note. So I guess I could conceive of much larger data sets. Actually, these performance differences for runtime and main memory usage, they could indeed become a bottleneck. And so in theory, we might need to think of more sophisticated techniques to deal with, I guess, computational tractability for larger data sets. In summary, what I've shown you is that basically we've run a simulation study and also other experiments where we validated that this random walk-based resampling and reestimation technique, it actually offers pretty important type 1 and type 2 error relative to state-of-the-art methods, at least for one application being multiple sequence alignment support estimation. Since the initial publication of this study, we've actually looked at a few other applications within just biomolecular sequence tasks. So we've looked at another classical problem being trying to detect recombination breakpoints, and indeed, random walk resampling helps there. In addition, trying to detect gene flow and quantify it within genomes. We've also run experiments showing that this technique can also be useful there as well. But honestly, kind of the reason why I'm excited to talk to y'all today is I actually think that much like the Bootstrap method, it's this whole idea, we could just be scratching the tip of the iceberg is maybe the way to poorly phrase it. So there's like a ton of other stuff that I'm really excited to apply this stuff to, and actually that my lab is working on right now. So I mentioned that my former student Hussein, his brother's working with me now. He's actually working on an application of these ideas to another topic that we care about deeply, which is phylogeny reconstruction. And we're seeing actually even better benefits there. And I'll briefly mention also that some of these ideas appear in some form in the machine learning literature and also in statistical inference and learning. And I have to wonder if some of these ideas could also be useful in that domain as well. So kind of for this, I'm kind of inspired by what's already been done by kind of, in such fundamental approaches as the Bootstrap method. And just because they're so widely used throughout really all of science and engineering, I think, I'm pretty hopeful for the promise of this particular approach. So if you're interested in finding any of the data and software for this method for your purposes, you could, in theory, look at our lab webpage for where we try to put all of our open data and open source software, and we try to put all of our data, published data, published software under open licenses. You'll find it at this particular URL. I will caution you that it's really research grade software. At the moment, we're trying to invest much more heavily into user-friendly and developer-friendly software and tools development. And so my student Wei Wang, one of the things that she's working on this year is putting together a really comprehensive package with nice documentation, nice tutorials, and we're hoping to publish that in a software paper soon enough. So we'll try to put a link on this webpage as well to point you to that once that appears. So we're working on better tools, better tutorials, better documentation. For the moment, though, if you're interested, you could take a look at this. I want to acknowledge, of course, Beacon for really an amazing, it's one of the reasons why I came here, honestly. So it's just an amazing center, amazing set of people, most importantly, and I'm really, really forever indebted to Beacon for many different things, but in addition to funding. So it's just been an incredible center to be part of, and I'm very privileged. Of course, I'd also like to acknowledge the NSF and MSU for also supporting the work that I talked about today, and in addition to all the other work going on in my lab. And with that, I'd be happy to chat with y'all about really anything about this research, about anything connected to it. I did include the link to the most recent journal publication for this project. And you'll find also the links for my lab website and my personal my professional website reproduced here as well. Thanks very much.
All right. Thank you so much for that great presentation, Kevin.
Sure, my pleasure. Yeah, thanks to everyone.
If anybody has any questions, I think we're a small enough group here that you could just unmute yourself, or you could type in the chat, whatever you prefer. So far, all I'm seeing is compliments for you, no questions.
Okay.
Yeah, I have a question.
Sure.
I'm just wondering if you've tried this with different kinds of data sets. So I do a lot of phylogeny reconstruction, and some things like ribosomal RNAs have lots of gaps and difficult ways to align. And some proteins like transcription factors have certain parts that are very alignable, but then the other parts aren't. And I just wondered if you've tried some of those different kinds of data sets, and how does this work on those different kinds of genes?
Yeah, thank you for that question, Billy. By the way, I've been following your lab's work for quite a while, but I never got a chance to introduce myself. So yeah, very nice to meet you.
Yeah, it's great to meet you too. I know your stuff too, so yeah.
Yeah, that's a really perceptive question. Short answer, I guess the most minimal thing that we've done is within that 2020 paper, we actually did look at the empirical data actually comes from the comparative RNA website. And so that benchmarking data is ribosomal RNA data.
Okay.
Yeah, it comes from the Gutel lab, where they've spent a lot of time curating these wonderful, very pretty much gold standard alignments, right? And so that's why we chose them for benchmarking purposes. But I think your answer is way deeper than that. I think there's a ton more that can be done for both doing the resampling better to take into account different features within your sequence data, right? Even just in the case of ribosomal RNA data, we included basically both in kind of the intronic portions of those loci as well as kind of the non-intronic portions. And so you see improvements on the intronic portions, but less so on the non-intronic. And so both the resampling and the reestimation procedures, they have no concept whatsoever of, well, anything beyond just the primary sequence data. So they don't know anything about structure, nothing about any of that. So all of that could be done much better. I totally agree. And so yes, both for the random walk resampling procedure and in fact for the reestimation, if you could incorporate more aspects of your data, more features of your data, like structure, that's just in the case of, I guess, RNA data.
Yeah, that's what I was thinking. Yeah, like something like ribosomal RNA, where some of the structures are known and things. But yeah, very cool, very cool. I look forward to having the tools to try some of this ourselves, maybe.
Yeah. I'd be happy to follow up with you. Once that paper comes out. Yeah.
Mm-hmm.
It's one of those things where, especially when I'm starting out, we just didn't have the time and resources to invest. But hopefully now I've got enough people, we can finally take the time to put together better tools. Yeah.
Cool. Thank you very much.
Sure.
Enjoyed your talk.
Thank you.
All right, do we have any other questions? Well, if not, I will thank you again and wish everyone a very happy winter break. I'm certainly looking forward to it.
Happy holidays, everyone. Yeah, I am too, I got to tell you.
Yeah. Have a great break, everyone.
Yeah. And I will have this recording posted in the next couple of days.
Cool. Thanks so much, Daniel.
Great. Thanks a lot, Kevin.
Yeah. Thank you all. See you.